didn't talk about it much last time. Surface area to volume ratio is just a math thing. Surface area is on the top of the fraction, volume is on the bottom. Cells want to have a high surface area compared to their volume because they take everything in through their surface. And if you have more surface compared to the volume that's on the inside, you can take more stuff in. So what cells will often do, instead of being shaped like that, spheres have the lowest possible surface area to volume ratio, by the way. That's the most volume you can fit inside any shape, a circle. That's why the Earth is round, because gravity pulls in and it forms something that has a lot of volume. So, what a cell might do is something like this. It might shape itself like this.
Whoa now. I've never seen
Doesn't that add a lot more surface?
Oh, it does.
Now it could also divide in two and make two smaller cells. That'll increase the surface area. I mean, you can imagine if this were divided into millions of small cells, you would have the same amount of volume, but way more surface. Because that would all be surface, and that would all be surface, and that would all be surface. If you could take this entire volume and put it into a bunch of small cells, it would be more surface. So you want a lot of surface area, you want less volume if you're a cell. What they're going to do is they're going to give you math problems on this. They'll say, let's say you have a cubic cell, 2 centimeters on each side. Cells are never actually that big, but we can pretend. So we have this cell. I'm trying to draw 3D. Is that any good? We have this cell that's 2 centimeters up, 2 centimeters sideways, and 2 centimeters deep. Find out its surface area to volume ratio. Would you be able to do that?
Yeah.
Are you good enough at math?
No, I can try.
What's the surface area?
6 times 2 squared.
There's 6 sides.
24
So it's 2 times 2 times 6. So 24.
Oh.
Do you mind that tag?
What's the volume?
8.
2 times 2 times 2. It's 8. So the surface area to volume ratio for that cell is 3 to 1. 3 to 1. You just reduce it. Right?
Oh, that was a question I missed. Yeah. About math. Oh, no, I didn't.
And they have
So let's say the cell's bigger.
Oh.
Let's say it's not 2 centimeters on a side. Let's say it's 4 centimeters on the side. Now the surface area volume of a 2 centimeter per side square cube was that. What's the surface area volume of a 4 centimeter per side?
Well,
How do you figure that?
You gotta do 16 times 6.
4 times 4 times 6. 16 times 6. State championship math team, 96.
Yeah, I was never.
What about 4 times 4 times 4?
64.
16 times 4 is 64. Good.
Oh my gosh, look at me go.
96 over 64. What's 96 over 64? State championship math team, 1.5.
So,
1.5. So notice it's 1 point. You can say
That's the same thing.
You can say 1.5 to 1. Notice that when it was 2 centimeters on a side, the ratio is 3 to 1. When it's 4 centimeters on a side, the ratio is 1.5 to 1.
uh-oh.
The bigger the cell, the smaller the ratio.
But you
You want a big ratio. You want your surface area volume ratio. Cells have it at like 4,000 or 5,000 to 1. Because they're real small. You don't want a low surface area to volume ratio, you want a high surface area. So this cell, 2 centimeters on the side, is better than this cell, 4 centimeters. Does that make sense?
It does.
You'll have to do a math problem like that on the test probably. They'll give you a common shape. If they did a sphere,
Oh no.
they would give you the formula for the volume of a
Okay.
sphere and the surface area of a sphere.
That's
Does anyone know the volume of a sphere formula?
I thought the volume was like 4/3
Have
something.
pi in it.
4/3
pi r
4/3
squared.
pi r cubed is the volume.
Cubed, oops, my bird.
And the surface area, you would get 2 pi r is the circumference times the height or something like that. Is that how you do it? Circumference times the, I don't know. Anyway, they'll give you the formula. There's a formula sheet. There's an AP Biology formula sheet.
That's great news.
That's so
What does
wacky.
it look like?
I don't know.
Well, I'll go to my AP Bio formula sheet folder, and it looks like this. You could just, if you Googled AP Bio formula sheet, it would come right up. If you ever need it, just Google it. And on the back here, there are equations. Look at this: volume of a sphere, 4/3 pi r cubed. Surface area of a sphere, 4 pi r squared.
Incredible.
Unbelievable.
Amazing.
Incredible.
Are you with me?
I am.
Can we do well on this test?
Yeah, I
Yeah.
think so.
We can. You have the ability. The AP Biology exam at the end of the year.
We have the technology.
Good, Thomas.
That's what I'm talking about. All right. Y'all ready for cells?
So ready.
I'm ready for you now. Heck yeah.
That sounds good.
Heck yeah.
Can you play the cell song?
Oh, yeah.
There's a cell song?
Yeah, the cells are made of organelles.
I will play it while we're working at our magnets boards.
Okay.
Yes.
So you can see I have cell part magnets. You got to be able to identify each part and tell what it does. That's what we're going to work on. I'm about to put you in groups. You go up there, you point to that, and you say what?
Chloroplast.
It's a chloroplast. What's it do?
Makes and does
Does photosynthesis. Makes food. What's it make food out of?
no.
Sunlight.
Sunlight. Not just that.
I was so confident. Oh,
And-
oxygen.
Water.
No, it makes oxygen. Carbon dioxide.
Carbon dioxide.
Oh, Josie, we just killed that. Let's go. Carbon
If y'all were on the
dioxide.
same team, you could have combined to get it right.
Really?
Hmm.
Dang.
Okay, we did all this. Hey, oh, there's me, the electromagnetic.
Mr. Willis.
Class yesterday.
We've been waiting.
How cool is that?
So cool.
When was that?
us that freshman year. Oh, I did?
Yeah.
That was in Nebraska, I think. I was running a workshop, and one of the ladies in my workshop was like, I run an electron microscope. And I was like, no way. I said, I've always wanted to use one of those. She's like, come on, I'll show it to you. It was in the university where I was giving the workshop. She took me in there. I sat down and had no idea what I was doing. But
Amazing.
pretty cool.
Did you look at something?
Yeah, she put a sample in and we looked at it. And it was pretty cool. Like you could scroll, it had dials, and you could turn the dial, and if you turn it far, it would scroll through real fast. And if you turn it short, it would scroll through real slow. And you're just watching all this stuff come up. And she's like, ooh, that's a so-and-so molecule. Ooh, that's whatever organelle. And I was like, cool. And so I scrolled. I was just scrolling right. It was going right and right. And it turns out I had gone through like 4 micrometers
Wow.
in like a half an hour of looking. I mean, it hardly moved.
That's a long time.
It focuses in so close that it takes a long time to see. It's pretty neat. It's fun. Okay, so anywho. Okay, here's all the parts of the cell. So we got to know all these parts. And so you got the membrane on the outside. So you'll come up here. I want you to draw a cell membrane. And let's do it right here. This. Let's move all this like this. There's a membrane on the outside of the cell. What's the membrane made of?
Phospholipids.
It's a phospholipid bilayer, that's right. Every cell has a membrane on the outside. Now, plant cells have a cell wall around the membrane. How would I draw the cell wall? Oh, I don't know. Maybe I'll use a green marker just to show that it's a plant. And the cell wall is usually pretty thick. And what's it made of? In plants, it's made of cellulose, in plant cells. What's it made of in fungal cells?
Chitin or whatever
In
it's called.
fungal cells, it's made of chitin. What's it made of in bacteria cells?
Peptidoglycan?
Peptidoglycan.
Yeah, Lottie.
Different
Vesicles.
organisms. Didn't you get that in science ball? Did you?
I don't remember anything from school, to be honest.
So different organisms have different cell walls made of different things, but that's just kind of a tough layer that holds the shape of the cell, and it protects the cell, and it only lets small things in. Water goes right through it. It's kind of like a chain link fence. You could spray a hose right through a chain link fence, right? Wouldn't stop much, but you can't run through the chain link fence.
I've tried it. It just does not work.
So that's kind of what the cell wall is like.
Sorry.
What else? Okay, we have the nucleus. That's the nucleus. What's it do?
It holds the DNA.
Holds DNA. Now, if I put a nucleus in it, it's obviously a eukaryotic cell.
Yes.
Eukaryotic cells have nuclei. Prokaryotic cells don't. Prokaryotic cells just got DNA floating around in the middle.
Just hangs out.
We'll make a prokaryotic cell next week sometime. I don't have the magnets here yet, but I've got them. Just don't have them with me. So a nucleus, that's the nucleolus. Nucleus, nucleolus. And you have to be careful. The nucleus is the whole thing. The nucleolus is this little thing here. That holds RNA. And that's where ribosomes are made. Ribosomes are made in this area. And then they exit through these little holes called nuclear pores. A lot of kids will write this when they mean that. If you write this in an essay, when you mean that, they can't give you the point. Because it's actually a different thing. It's like writing cow when you mean cat. It's a different thing. Hold on for a minute. Charlotte, what's up? Sarah? Okay. Does a one-legged duck swim in circles?
Heck yeah, it does. Yeah.
I got 40 yearbooks right
Oh,
now.
thank you.
But you got to return them because
We will.
they go all the way back to my childhood.
Mason, what
Heck
year
yeah,
were
it's
you
on
born?
here. Why don't you have your mask over your nose?
Ooh, silly.
Do it, Mason.
He's not even. Which years did you born?
Easy. All right, so we got this. So this is the nucleus, the nucleolus, the nuclear membrane. The holes are called nuclear pores, and the little brown dots on it are ribosomes. There are some ribosomes on the nucleus itself, and you can see the nucleus in the picture here and here. Okay. Then I'll put these things. These are what? Do you know?
Endoplasmic reticulum.
Endoplasmic reticulum, ER. Now there's two types of endoplasmic reticulum. There's rough ER and there's smooth ER. And it looks different in different cells. It's usually close to the nucleus. So there's the nucleus, there's the ER. And here it is down here, nucleus, ER. Now it can be far from the nucleus also. Some cells are just full of ER. Almost the whole cell is full of ER. Your liver cells are like that. Because they have to make, if you have a lot of ER, you're making a lot of stuff. Because rough ER are where proteins are made, and smooth ER is where lipids are made, fats. And they're all put together in these locations here. And so, you're welcome. After the rough ER and smooth ER, rough ER has ribosomes on the outside of it. Smooth ER doesn't. Y'all remembering any of this?
Yes, sir.
After they, God, this is the tiniest let me get a bigger one. After they make the proteins or the lipids, they ship them off in a vesicle. A little bubble that holds stuff. So here's a vesicle that can ship things off, and usually it'll be shipped off to the Golgi body. This is a Golgi body, named after the guy who discovered it. The Golgi body will take the proteins and the lipids that arrive to it, and it will change it. There are enzymes in this thing. Layers of enzymes that will make changes sequentially, almost like in a factory, like a what's it called in a factory that has different stages?
Assembly line.
An assembly line. It's almost like an assembly line. There's enzymes in that packet and enzymes in that packet. It's like many layers of flattened sacks, stacks and stacks of membranous sacks. I will not eat green eggs and ham. I will not eat them. Tom, I am. So, these stuff gets shipped off, it goes through the Golgi, and it moves through it. Oh, I should have had this on. I screwed that up. This is supposed to be on. Have you ever seen, like, how material moves on a lava lamp?
Yes.
Yes.
How it kind of loops up, and just kind of, and if two bubbles hit each other, they join together.
Mm-hmm.
That's how material moves in the cell. These bubbles, this bubble right here, will become part of the Golgi when it runs into it, just like lava lamps do. That's why I got the whole lava lamp in the first place for this class, and I forgot to turn it on. So anyway, okay, so that's there's Golgi. There's usually several Golgis around cells. Some cells have mini Golgis, some cells just a few. There's one there. There's one there. Then we've got what else? Ribosomes. Sometimes ribosomes are attached to the ER. Sometimes they're floating freely in the cell. The free-floating ribosomes will make proteins as well. The ribosomes that are just floating around in the cell, they make proteins that stay in the cell. The ribosomes that are on the ER, they make proteins that end up in the membrane of the cell.
Whoa.
So they're on the membranes here, and they end up in the membranes of the cell.
Is that like the cholesterol that's in the cell? Or is that
Cholesterol does end up in the cell membrane, and it is manufactured in the smooth ER.
Dope.
Yeah, that is correct. What else? We've got, there's other types of vesicles out there. There's big vesicles that contain digestive enzymes. We call them lysosomes. Have you ever heard of that?
Yeah.
I like to call them floating balls of death. Because if a bacteria or something invades, the lysosome can be taken by the cell to a lysosome and be digested there. This will digest almost anything. Cells can actually kill themselves by making a bunch of lysosomes and then breaking the lysosomes apart in the middle of the cell.
Do they do it on purpose?
They'll do it on purpose. It's called apoptosis, which is also known as cell suicide. And your cells, if they turn cancerous, usually they'll kill themselves. We've all probably had cancer thousands of times already.
That's
But the
wonderful.
cell killed itself. So we're just fine. The problem comes when the cell becomes cancerous and won't kill itself.
I can see how an issue would come out of that.
Yes. So we have, there's other vesicles, peroxisomes you'll read about. They tear apart hydrogen peroxide. Hydrogen peroxide is a poison that builds up in cells. It can be torn apart in the peroxisomes. And then there's little vesicles. There's big vesicles, there's little vesicles. Sometimes if it's a plant cell, you'll have a big water vacuole. This one, the water vacuole's there. A water vacuole is a larger vacuole that holds water. Plant cells need a lot of water. And often the water vacuole will be way bigger than this. It might take up half the cell. And the water is used for photosynthesis. I'll put some chloroplasts in here. They do photosynthesis for the plant cell. They aren't in animal cells, but you find them in plant cells. All cells have mitochondrions.
Yeah.
Mitochondrions take glucose or other simple sugars and they turn it into energy. Now, of course, at this scale, this molecule would be so small you wouldn't even be able to see it. It would just be a little dot. And these glucose molecules go into the mitochondrion, and the mitochondrion breaks them down to make ATP. We spent a whole chapter learning about how that works. We spent a whole chapter learning about how that works.
Amazing.
That's like a week and a half of class.
Is that like the citric acid?
Yes, yes. Citric acid cycle happens in that. And this is a Calvin cycle happens in that. I don't know if y'all remember that. That's two of the hardest chapters. They're right in a row.
Oh,
Bam, bam. Not next test, but the one after.
wait, what?
That's when we study those.
Yeah.
It won't be on the next test. It'll be on
Is
the
that what
one
makes
after.
ATP?
Yeah, that makes ATP. And that makes glucose. What else do we got? Well, cytoskeleton. That's this stuff right here. Are like little train tracks that run through the cell. In actuality, these vesicles, they transport themselves along these tracks. They don't just float around randomly. They're usually stuck to one of these tracks and they move. Okay? And so that's called cytoskeleton. The cytoskeleton can also make make the cell shaped a certain way. It can cause the cell to be like long and thin if you have a bunch of long, thin cytoskeleton running this way. Those are proteins in the cell that give it a certain shape, give it support, and allow things to move. So the last thing, these are some centrioles. They're only in animal cells. You don't find them in plant cells. They're active in cell division when the cell divides. When we do mitosis and meiosis, I don't know if you remember, these things went to opposite sides of the cell and shot out spindle fibers. Do y'all remember those?
Yeah.
That's these things. So more about that in later chapters. And then we can throw some cilia on the outside of the cell and maybe a big flagellum if we wanted it to swim around. The flagellum wags back and forth to push the cell along. And the cilia kind of moves like that to pull fluid along past the cell. And maybe that's how it eats, just depending on the cell. So here's all these parts. I want you to go with your biomedia groups to a board. Hold on, don't get up yet. And what you're going to do is you're going to draw a cell, and you're going to take all the organelles, put them in one by one, and say what it is and what it does, and then let the other person in your group go. And see if you can get them all right. And if you can do that, you're doing pretty good.
Wow.
Okay? You can turn that off if people don't need to see it.